So I will share my screen. And what you see now, hopefully, is the booking system. So that's where you can book your time slots for using the equipment at the facility. And every new user, the first step to become a user is to create an account in the booking system. Our facility is a part of Singascope, which is a network of microscopy facilities in Singapore. There are facilities at A-STAR, NUS, participating in Singascope, and the booking system is common for the whole Singascope. So if, let's say, some of you have used any other of the Singascope facilities, in that case, you already have an account in the booking system and you don't need to create a new one. I'll start with home screen. So the home screen for you might look a bit different because there might be features which are only relevant for admins. So I am now logged in with my account, which is an admin account, but some features which everyone should see is here the selection to book a microscope. So you will, here you can see which microscopes are available. Now this is the complete list of all equipment in the facility. There's equipment at Celsi, and because our facility is a joint facility with LKC Medicine, so you can also see equipment at LKC Medicine. For most of you, the list will contain less items because there will be only those microscopes which you are allowed to book. So that means the microscopes which don't require training, that means those simplest ones, or microscopes you have been trained to use. And if you want to use a microscope you haven't had the training for, you can request a training here, which will direct you to an online form. This will be the online form, and there you will be asked questions about your intended experiments, if you know which microscope you need to use, or if you need our advice, and also if you need any special conditions for your sample during the imaging. So I won't be filling this in now, and I will return back. So then you can also see the schedule where there are all the equipment in the booking system, and you can see when the equipment is booked and when it is free. If I click on any of the systems, let's say here this one which has some bookings today, so then you will be directed to the calendar for that particular microscope. And you can also create your bookings here in this interface. So if I would want to book, let's say, I'll move to next week, and then I can make a booking by selecting the time which I want to book. Yeah, there will be, of course, your name, and your default project. So some the projects allow you to use, let's say, if you are working on multiple projects, then this will allow you to link particular bookings to a particular project, and this is relevant mainly in terms of payments for the microscope usage, because then the sessions can be paid from different grants, depending whether you are working on one project or another project. And then I would press here, book selected sessions. And that will place the booking into the calendar. So let's do it, and you will see that the booking is there. There might be, of course, situations when you need to cancel your booking because, for example, you need to urgently do something else in the same time. You can cancel the booked slot. So if I click on it, then I can do some changes. For example, change the project, but I can also cancel session, and session is gone. You can cancel it anytime before the start of the session. So if the session was about to start at 10:30 on Monday, anytime before 10:30, so 10:25 on Monday, I can still cancel the session. So in that sense, it's very flexible. There was a discussion in the past whether there should be any cancellation limit, because some facilities impose a cancellation limit, so you cannot cancel your bookings within some interval before the start of the booking, or you can cancel, but you still need to pay for the booking. But then the conclusion was that either it becomes very inflexible, the system, or if we impose a very short cancellation limit, like let's say 1 or 2 hours, that doesn't really help anyone, because if someone cancels 2 hours, that's still doesn't increase much the chance that someone else will have a sample ready to utilize the slot. So for that reason, we decided we just don't impose any cancellation period, and simply you can cancel any time before the start of the session. You can cancel even within the duration of the session. So that means if the session started at 10, was a session from 10:30 until 12. So if I cancel it at 10:35, I can still cancel the rest of the session. I cannot cancel the whole session, but because you see the calendar, the booking is in 15-minute slots. So if I cancel it at 10, I booked from 10:30 and I cancel at 10:35, I will pay only first 15 minutes. Because it works in 15 minutes step, but I can cancel the rest. So only what will remain are the first 15 minutes of the session. I have to illustrate this better, I have created a slide. So I can now go maybe to. So this is so this could be this represents the calendar, the sessions, and we have two users. One user is shown in blue, the other user is shown in green color.
That we can't see your other screen.
You can't see.
Yeah, it's still in the browser, the PPMS
Okay.
browser.
Okay. Let me.
Okay, you
Okay.
can see it. Yeah.
Okay. Thanks, Young. So now you can see the slide. So there are so these tables represent the calendar in the booking system, and the different colors represent two different users. So if a user A books these slots, and then the user use these slots. So because on each of the computers at the microscope, there's also a usage tracker, which is basically a program where you need to log in with your PPMS account, and that is linked to the PPMS booking system. So in this way, the booking system knows when which user is using the equipment. So if you are using not within your booked slot, but in a slot which is shifted, then you will have to pay for uh, the whole time, which comprises of the booked and the actual usage. So whichever is longer, you will have to pay. If you book a long slot, and then instead of you, another user B, instead user A booked, but user B is actually using the microscope. So then in that case, user B will pay for the time when user B is using the microscope, and user A will pay only for the excess of the booked time. If there is no booking, but user A is using some time slots, then user A will pay for those time slots. User B is using different slots, so the user B will pay for different slots. I wanted here by the one cell in the table would represent the 15 minutes, which is the precision of the booking calendar, but the usage tracker works on minute precision. So here, if the user is using, let's say, 8 minutes of this 15-minute interval, they would pay only for the 8 minutes, not for the whole 15 minutes. And if a user A booked a long session, let's say to make a time-lapse experiment, and then they in first 15 minutes they found that there's something wrong with their sample, so there's no point in doing the time-lapse. So they shortly after that the user cancels the session. So then the user would pay for let's say this is the 15 minutes, so canceled 20 minutes after the beginning, so the user would pay for 30 minutes of the time. So I hope I didn't make it too confusing, and it is understandable how the booking and the payment work. So of course there might be situations when you have an emergency, like your lab is flooded or something like that, and instead of going to measure at the microscope, you rush to, or your freezer stopped working, so you are rushing to save your samples, and in that case you will maybe maybe you forget about, or you don't have time to cancel your session. So in that case, of course, we don't want to charge you for the unused session. So if it's a well justified reason why you couldn't cancel the session, so you can always write to Yongvi or to me, and we as administrators can cancel the charge for the session. And of course, you don't expect that you will be having an emergency every other day. So this is something which is, of course, which is we don't expect to be happening very frequently. But of course, it's no problem to adjust for that. So I will go back to so can you see now again the booking system?
Yeah.
Okay. I believe that this is this is basically all I wanted to say about the booking system itself. Now let's move to, let's say, information resources about our microscopes. So in the booking system, you can see, oh yeah, okay, one more thing here, you see the charge rate NA is because I am logged in with my project. So I'll change my project. And now this is what most of you will see is the charge, the rate, the per hour rate of the instrument. So for this particular wide field microscope, it's 8.4 Singapore dollars per hour. And for all microscopes, there is a long-term discount. So if your experiment is very long, like long-term time lapse, then it might be very costly if the whole time is charged at the same rate. So after 8 hours of measurement, so 8 hours will be paid by the standard rate, and after 8 hours, 75% discount applies, and after 24 hours, 95% discount applies. So if you do a time lapse which extends over several days, the price will not be that high. And here you have more information. So if I click this, it will bring me to a page about that particular microscope, which contains some basic description and details of objectives, fluorescence filter set. We will talk about these things later, and how, if in case you are not familiar with microscopes, then you might not understand so well all these parameters here, but we will talk about that later so that you know what all these things mean. And there are some there are other features which characterize this microscope, and so this is what we call system overview. And another thing for, well, not yet for all microscopes it's finalized, but for at least some of them, we have a manual. which describes how to use the microscope. So that's basically, those are the things that we will tell you during the hands-on training. So how to switch on the microscopes, how to switch it off, how to acquire images in transmitted light, fluorescence imaging, multi-channel imaging, and time-lapse imaging. This is a manual which describes basically what we would tell you during the training session. So in case you you have had a training session for a microscope, but maybe haven't used it for a long time, you can also use this as a reference source to catch up on how to use the microscope if you are not sure about something. And of course, it contains a link back to the booking system for that particular instrument. And for some of the microscopes, we have a table of excitation intensities, which is basically what is the excitation intensity for each objective and for different filters or different lasers in the case of confocal microscopes. And there's also a link to the website of the imaging facility, which we can have a look at now. So the more interesting parts of this website are a list of microscopes. So if I press any of these, so it will bring me back, will bring me to the system overview page for that microscope. And there is a link to the login page to the booking system. So for new users, you can follow this link to create a user account in the booking system. And there are, we put here links to training materials from Microscopy Australia. They have quite a nice training materials called MyScope. So I will mention later a few other sources of information if you want to look at more at theory or at study a bit more about optical microscopy. And one important thing here is there's a mention of Omero. So Omero is an image repository. It runs at a server at CellC, and you can use it to store and archive your images, and also to transfer images from the microscopes to, let's say, to anywhere else, because we don't allow any external hard drives or any USB thumb drives to be used at the computers in the facility. So there are two ways how you can copy images which you acquire at the microscope. One of them, which is available for all users from CellC, is Omero. So when there is any new user of the facility from CellC, we create an Omero account for them, so they can log into the server and upload images. There's also have a look. This is how to use a very brief guide how to use Omero, how to uh, how to upload images there, and you can store your images there. You can, of course, connect to Omero from any other computer in on campus. If you want to connect from outside of campus, you have to use VPN. But you can, so even from outside of campus, you can connect to the Omero server, and then you can view your images or you can download them. And another way how you can transfer your files is mentioned also here in this guide, and it's a shared drive, which is called which is mapped as drive Z at all the microscope computers, and you can you can copy your files to this drive, and again you can map the same drive at your PC. The same you can access it from any PC on campus. If you are off campus, you need to use VPN, and then you can access the files, and you can copy them from the shared folder. This is there's a note that this is meant for, unlike Omero, where you can archive your images and keep them for ever until the collapse of civilization or the failure of the server, whichever comes first. It should be the server should be backed up somewhere, so it shouldn't depend on the server alone. So you can this is meant only for data transfer. So whichever files are longer there than 15 days, so after 15 days the file is deleted to prevent accumulation of too many data and flooding this disk, which is not that big. So, and of course, another thing, unlike Omero, where you need to log in to access it, so there's a control what files each user can access. So this is a simple shared drive. So whatever you put there is accessible to anyone else. So if you have any files which contain confidential information, this is not a good place to put them. Okay, so this is about Omero. I think this is, so this there's probably not much more you can find here. I just recently added one more link here because that seemed to be quite in demand. So this is a link to apply for card access in CellC, because there's a card access to the facility as a whole, and then to the rooms which house some of the microscopes. So once, if you want to become a user of the facility, you will need to get card access there. So this is again a link where you can access the form which you fill in, and where you specify which door you will need to get access to. Did I forget anything important? Maybe Yongri, if you want to add something.
I think you cover everything.
Okay, so maybe we'll find out something else, but for now, we can probably move to the next topic, which is the, so I will switch now to sharing the PowerPoint presentation. Can you see it?
Yep.
Okay, so we saw this slide already, and this is actually the title slide of this presentation. And so first I mentioned here a few resources which might be useful for people doing microscopy. And so one of them, which is something what I've already mentioned, it is called MyScope, and it was created by Microscopy Australia, which is something like Singascope, but in Australia. And it contains training materials which explain the theory. There are some interactive applets, and also there are some quizzes which you can take to check whether you have learned anything from the materials or not. So this is a very nice thing. Another, which is a very classical source, I would say, is called Microscopy Primer, and this is it's quite an extensive website which contains a lot of microscopy related things. It was started by late Michael Davidson from Florida State University, and it's a textbook which explains all sorts of things in optical microscopy, the construction of an optical microscope, operation, um, different modalities of optical microscopy, and also it contains galleries of microscopy images. Um, there are articles on the history of optical microscopes and other fun stuff, not really that related to actual usage of microscopy. Another thing is on iBiology website, they have a microscopy course. So this is it's a series of talks, of presentations which explain how to optical microscopy for biological applications. A similar thing on Endor website. So you might know Endor as a company who manufacture scientific cameras and also spinning disk microscopes. So they also created a course, an online course on microscopy. I forgot to mention that this microscopy primer, so there are derivatives from it, uh, which because this was, I mean, at its time, that was something which was quite unique in the extent of the all the microscopy related articles and explanations it gave. So a lot of microscopy companies have derivative websites which use the articles from Microscopy Primer. So there's a website called Microscopy U, by Nikon. There's Zeiss campus, obviously by Zeiss. I forgot what Olympus called that, but they also have a derivative. So they are actually very, those are all very similar, and you can find that they are using materials which are all come from microscopy primer. Obviously, when there are pictures of a microscope objective, so if you look at microscopy U, it will be a Nikon objective there. If you go to Zeiss campus, it will be a Zeiss objective, but otherwise the rest is practically the same. Zeiss have now created a new, they are basically, the Zeiss campus is still somewhere there online, but they are not supporting it anymore. So they have a new website for all the microscopy related resources, which they call Zeiss Academy. But unlike Zeiss campus, you need to create a Zeiss account to access it. It doesn't cost you anything. You just need to sign up for it. So you just need to register somewhere so that they can send you emails, promotional emails, probably. And a bit different thing is FPBase, which is a project, a database of, it started as a database of fluorescent proteins, but and it contained a spectra viewer, which showed emission and excitation spectra of the fluorescent proteins in the database. But the viewer now contains also a database of not just fluorescent proteins, but of any fluorophores. So there are other, of course, there are also other databases. I just mentioned this one because I personally, this is probably my favorite one. So if you want to see what is the spectrum of your fluorophore, this is a resource where you can find it. I will later show you some, I can show you later some spectra which were taken from this database. Okay, so now let's talk about the microscopes at the facility. We can divide them into two categories. Electron microscopes, which consist of a single instrument, a scanning electron microscope, and optical microscopes, which is all the rest. So this is just an example. I couldn't fit the pictures of all of them here. But of course, these pictures don't show much. So let's mention more about the underlying principles. So in electron microscopy, as the name suggests, and in scanning electron microscopy, we have a focused beam of electrons which are interacting with the sample, and we are looking at either electrons from the beam which have been scattered from the sample, or, and that's typically the main source of the signal, are secondary electrons. So those are electrons which have been, so when the primary electron beams hits atoms and molecules in the sample, it can kick out some of their electrons from their orbits. So those are the secondary electrons which are detected, which are then detected as the source of signal. One thing, because electron beams don't travel too far in any material, so it has to be all done under vacuum. And if you put water into vacuum, what will happen? It will immediately evaporate, and that means there's no more vacuum there. So you have to work with dehydrated samples. So the samples cannot contain any water. And to prevent accumulation of charge when you are hitting the sample with electrons, the sample needs to be conductive so that you can drain the excess of the electrons from the sample. And on top, the best contrast comes from heavy metals. I'm not talking about any music genre here. I'm talking about things like gold, platinum. And for that reason, the samples are typically coated unless you are looking at a piece of metal itself. But if you are looking at something of biological origin, then it's typically coated with gold or platinum, a thin layer of those metals. So obviously this is not compatible with anything living. On the other hand, optical microscopy can be done under physiological conditions. So you can looking at living cells, living organisms. Another aspect discussed is the resolution in electron microscopy can be on the orders of nanometers. It can be better, but for the SEM, the type we have at CellSeed, it can reach resolution in units of nanometers, and it has an extremely large depth of field. You might not know what is meant by that. So I illustrated here by showing an SEM image of a seashell, and you can see from the scale that the depth means the height of the sample is on the orders of hundreds of microns. So I can easily see over the range of hundreds of microns, I can see the object sharply in focus. So the depth of focus is it can be very large for electron microscope. For optical microscopes, the resolution is about 2 orders of magnitude worse, and the depth of focus is also much shallower, depending on the type of optical microscopy we are doing it. But typically, we can't see anything like 100 micrometer can't be simultaneously in focus. I mean, can be both curse and blessing, because I can see a whole large sample simultaneously, each part of it in focus, but it makes it very difficult to do three-dimensional imaging in SEM. I mean, you can see that the image gives you a three-dimensional feel, because there are things like shadowing effects which serve as depth cues in the image. But to rigorously measure the height of an object in SEM is not trivial. While in optical microscopy, because of the shallow depth of focus, by just looking which part of the sample is in focus allows you to measure distances in the height of the sample, the axial distances. Now let's go to optical microscopes, which is that's where the majority of the microscopes in the facility. And we can divide them again into 2 categories. Based on the construction of the microscope, we can divide into upright. In an upright microscope, the objective is above the sample. An inverted microscope, where the objective is below the sample. So we are looking at the sample from below. Each of them is more suitable for a different type of samples. So for inverted microscopes, the typical samples, this is ideal for cells in a dish, because if we have cells which are sticking to the bottom of the dish, so it's very convenient to look at them from the bottom. On the other hand, the upright microscope is better for large non-transparent samples, because when we are coming with the objective from top, we can see better where we are looking at the sample. So typical examples are larger animals, such as rodents, like a mouse in this example, or if you have other if you want to see something on the surface of non-transparent materials, like if you want to see bacteria growing on a piece of stone, for example. Things like histological slides, which is basically you have some thin section of something which is squeezed between two pieces of glass, are suitable for either of them, but might be preferably observed under upright microscope, simply because in a facility like CELSI, where people do a lot of work on cells in dish, the inverted microscopes are otherwise in much higher demand. These are examples. So we have one upright microscope, and then we have two very simple microscopes which are also upright, and the rest, again, that's more of the microscopes are inverted microscopes. Then there are different modes of illumination in optical microscopy. One which is called diascopic illumination, or transmitted light illumination, which I think this explains quite well what it is. So that means we are shining light through the sample, and we are looking, so we are shining light from one side and we are looking what comes out on the other side from the sample. Now we have two lenses. One is called a condenser, so the light comes from the condenser, and then we have the objective lens on the other side of the sample, which collects the light that has passed through the sample. Again, this can be in both inverted and upright configuration. The other is called episcopic, or reflected light. And you see the difference is I have removed here the condenser lens, because in this case, the objective lens typically, I'm basically looking, I'm shining the light from the same side of the sample where I'm looking at it. So typically the objective lens work as the condenser lens as well. So I shine light through the objective, and then at the same time I'm collecting the light that comes back by the same objective. In our daily life, we mostly observe things in the episcopic mode, because most of the things which surround us are not very transparent. Things like furniture, buildings, human bodies are not very transparent to light. So the fact that when we are looking at a building, so we see the reflected light, we don't see much of light coming through the walls. Here, to give some examples. So this is obviously it's not a microscope image, it's an image taken with my handphone. So this is an image in transmitted light. This is our bathroom door, and there's no light in the bathroom. There was light in the kitchen. So I took this from the bathroom side, so I can see the shadows of stickers which my daughter placed on the wall, which is on the door, has this sort of milky glass. And now this is a reflected light image. I am on the kitchen side, which means I'm on the same side where the light source is, and I see the light reflected from the door. So let's look now at the transmitted light. So except of like looking simply at the light which passes through the sample, which is what would be called bright field, the thing is that unlike furniture and buildings, a lot of samples used in biology, like cells, they are quite transparent. So that allows us to see them in the transmitted light. But sometimes they are too much transparent. So they don't give too much contrast in transmitted light. So there are different ways how to enhance the contrast. And those are based not on the absorption of light by the cells, but more on the differences in the refractive index between the cell and the surrounding medium. So there are techniques like phase contrast, DIC, dark field, which is using scattering of light, oblique illumination, which is sort of poor man's DIC, and polarized light, again, if that's especially can help to visualize some oriented macromolecules which depolarize light. So basically, the bright field is the simplest thing. So on any microscope which can do transmitted light, you can do bright field. For all these things, you need some special components. For phase contrast, you need a specialized objectives. For DIC, you need to have special prisms which are inserted into the condenser and below the objective, and so on. Interestingly, these things which now might seem to us very routine, like phase contrast, so actually both phase contrast and DIC, the discoveries of them led to Nobel Prizes, similarly to more recently super resolution microscopy. So one thing you see here, there are two bright field images. One is a grayscale image, one is an image in color. And sometimes people come and ask if we have if they need to take an image, a bright field image in color, and that the microscope they are using has only a monochrome camera. There is a simple trick which I want to show here. So if you have a microscope which is meant for fluorescence, so that will have different fluorescence filter sets. So you can take a bright field image. Normally you take bright field image without any filters, but you can take also a bright field image with different filter sets. So you take one with a filter set which is transmits in blue, so let's say DAPI filter set, one with a green filter set, one with a red filter set. Then you basically take the images which are monochrome images, but you can give them relevant lookup tables, blue, green, and red lookup table. And then if I overlay these images, I get an RGB image with a monochrome camera. So if you want to use this, there's also in the manuals for the wide field microscopes, we have there's a section which describes how to do this trick, and there are also experiment settings which you can load and which will automatically switch the channels for you, and you can acquire easily a bright field image in color. So let's go back to, so we talked now about the bright field, the transmitted light. Let's go back to the reflected light. So this is another example of a reflected light image. Again, it's not a microscopic image, although this is something which is more biomedical, because this is, according to my daughter, it's an anatomy chart. So there should be liver, stomach, brain, all is there. Don't ask me what is what. And this is so this is normal reflected light, and this is an epifluorescence image, which is acquired when I illuminated. I switched. There was in a dark room, and I illuminated with a UV light source, something which is sometimes called black light, because you don't see the UV, but you see fluorescence from objects which are illuminated by the UV. So white paper is quite strongly fluorescent. So it has the bluish fluorescence. So you can see that the white paper glows, but the part which are covered by crayon, so you can't see their colors because there's no yellow or red in the light in the spectrum of the light which was used for the illumination. So this is and this is something which is very important, epifluorescence. And let's talk now about fluorescence and fluorescence imaging, because that's after all the large part of the work that is done in the facility and in overall in biological microscopy nowadays is fluorescence microscopy. So let's talk briefly about the phenomena of fluorescence. So to see fluorescence, so we see the fluorescence of certain molecules which are fluorescent, can be fluorescent proteins, organic molecules, nanoparticles, and that allows us to selectively label certain structures in the cells or organisms with a particular fluorophore, fluorescent molecule or fluorescent nanoparticle, and then we can selectively look at particular structures. Fluorescence has amazing thing that it allows us to see individual molecules. An individual molecule, unless it's a huge macromolecule, otherwise a smaller size molecule won't be visible in any transmitted light or DIC or anything like that. But in fluorescence, I can see individual molecules, which is something which is quite interesting. And here are the diagrams which show how does the what happens during the fluorescence. Don't worry too much. We don't need all the details of it. But what happens is that molecule absorbs a photon, so the energy of the molecules increases. It receives the energy from the photon. So the molecule goes to a higher energy state, and then the molecule. So this is connected. The increase in energy of the molecules is connected with different arrangement of electrons in the orbitals, and then the molecule can so it can go. There are different energy levels to which the molecule can be excited, but then typically very fast the molecule returns to level S1, the lowest. So these larger sets of levels labeled here S0, S1, S2. So they correspond to different structure of electrons in the molecule. These finer finer structure of energy levels corresponds to vibrations of nuclei. It's not important such details, but the important point is that the molecule almost immediately return to the lowest vibrational state of the S1 state, and that's where the molecule stays for some time. And it's from this state that the molecule then returns to the ground state by emitting fluorescence photon. So one thing which we can observe here is that the fluorescence has lower wavelengths than the excitation. Because I excite to some higher energy level, and then part of the energy is dissipated without in a non-radiant manner. So there's only part of the energy is then used for the emitted fluorescence photon. I mean, it is something which is quite intuitive. It's basically that is related to the energy efficiency of the whole process. So some energy is dissipated along the way. So not all the energy is then emitted back in the fluorescence photon. And the lower energy means longer wavelengths of light. So that means that the fluorescence spectrum is shifted to red from the excitation spectrum, and that is called a Stokes shift. Some fluorophores might have longer Stokes shift than others, and we will later mention that this is something which is very important and is used. This fact is used in the way how typically fluorescence microscopes operate and how we separate the excitation from the fluorescence. Now another thing which is also worth noticing here, that because, so you see that doesn't matter whether I excite by photon of such an energy which excites to the S2 state, or whether I excite just with enough energy to get to the S1 state, to the lowest vibrational level. The photon is always emitted when the molecule is at this state. So that also means that the fluorescence, the emission spectrum, is independent of the excitation wavelength. So you see, doesn't matter whether I excite here by such a wavelength or by this wavelength, the emission spectrum has always the same shape. Only the amplitude, of course, might change because different wavelengths will excite the molecule with different efficiency. But that's an important thing. So if there is any if the emission spectrum is changing with the excitation wavelengths, then this means there's something else going on. It can't be just simply fluorescence of a single fluorophore. And another thing is that it's very important is that fluorescent saturates, because the molecules spend always some non-zero time at this state, at the lowest vibrational level of S1. So the time is typically quite short. These are some nanoseconds, but it is a non-zero time. And while the molecule is in the excited state, I can't excite it again. So that means that when I excite a molecule, if there's any other photon coming in the way, it can't excite the molecule until the molecule returns back to the ground state. It's like if you give a child a new toy, so the child gets excited over it and stays excited over it for some time. So similarly, if I give a molecule a photon, the molecules get excited about it and will stay excited about it for some time. The time is, of course, quite short in this case, but it's still, and it has important consequences for uh, practical consequences for your experiments. So if I measure the dependence of fluorescence emission intensity on excitation intensity, so at low excitation intensities, it will be approximately linear because the molecules are mostly in ground state. So if I send more photons, I can excite the molecules more, I can excite more molecules or excite each molecule more frequently. So I have a gain in emission intensity. But then I reach the stage where the molecules are already too many molecules are in an excited state, so I don't have enough molecules ready to be excited. So sending more photons in doesn't help. So that means that when you are setting up your experiment, so it's better to start with lower excitation intensities, lower laser powers, and then increase that slowly and see if I have a corresponding increase in signal. If I don't have, then probably I am already reaching the saturation level, and increasing the intensity doesn't help. It just increases the risk of phototoxicity or photobleaching of the molecules. So it's better to be somewhere, don't go over the linear, don't go too much over the linear range of this dependence. Now the schematic of a epifluorescence microscope. We have a light source, which can be a lamp, a laser, and then there might be an excitation filter. In case I'm using a white lamp, then there has to be an excitation filter. In case I am using a monochromatic laser, I might not need one. Then there is something called a dichroic mirror, and that's where Stokes shift is applied in practice. So this is a spectrally selective mirror, which is designed in such a way to reflect, typically in such a way that it reflects the excitation light to the objective, and then the fluorescence, which has a longer wavelength than the excitation, will go through the dichroic mirror. And then there is typically an emission filter, which again allows me to select the range of fluorescence wavelengths I want to see, and then whatever passes through is detected. It can be either a spatially resolved detector, a camera, or it can be a photodiode, a photomultiplier tube. So now, of course, the selection of these dichroic mirrors and these filters, this is an important step in designing imaging experiment and has to be selected according to my fluorophores. So here, this is an example of spectra from my favorite FP-based spectra viewer, where I selected two fluorophores, fluorescein and DS red. And I can see, and here is a list of filter sets from one of our microscopes. So I can see which filter set will work for which of the fluorophores. So filter set 10, which has excitation between 450 and 490. So 450 is here, 490 is somewhere here. That seems to excite very well fluorescein. Then there is a dichroic mirror, which reflects below 510 and transmits above 510 nanometers, and emission filter, which transmits from 515. So that is 515 is somewhere here to 565. So That will allow me to collect most of the fluorescein emission spectrum. Similarly, this DS-red filter again will work very well for DS-red, which is not too surprising. Here you see a slightly different notation. So sometimes the filters are described in a range of wavelengths where they transmit. And sometimes there's this notation that there is the central wavelength slash the width. So I should understand this, that it transmits that the center is 545 and the width is 25. So that means 545 minus 12.5 plus minus 545 plus minus 12.5. You can see that if I excite that if I am exciting the fluorescein, I will also quite efficiently excite DS-red. So that's why it's in this case I might have difficulties because the range for the DS-red emission, for the fluorescein emission filter, will also allow some of the DS-red fluorescence. So it's sometimes if I have fluorophores with overlapping spectra, I might face issues that I have spectral crosstalk. That means that one fluorophore contributes to the channel for the other fluorophore. There are, of course, I can try to avoid it by selecting fluorophores which are more spectrally separated, or using narrower filters, or the other way how one can get around it is measuring in multiple spectral bands and then doing spectral unmixing. So if in case you have such problems, I won't be too much to explain now, talk about now about spectral unmixing, but if you think you need it, then you can approach us and we can help you to how to do the spectral unmixing, so you can get the signal from each fluorophore without the spectral crosstalk. So especially if you want to do things like quantify colocalization, then the spectral crosstalk is a serious issue. Other thing, one more thing. So traditionally, these emission excitation filter and dichroic mirror come in a mounted in one holder in a sort of filter cube, but sometimes at some microscopes there are separate turrets for excitation filters, for emission filters, and for dichroics. So that gives you more freedom. So you can combine them arbitrarily. So in case you have, for example, fluorophores with different Stokes shift, so you can use the same excitation filter but different emission filters. And in case of confocal microscopes, there's those more high-end confocal microscope typically come with spectral detection. So that means that I don't have filters there which will select a certain spectral range, but I decompose the light into individual wavelengths, and then I select which part of the spectrum will reach my detector. So either I can scan the spectrum over the detector, as it is done on Leica confocal, or I can have an array of many detectors which can capture the whole spectrum at once. So this is how it is done in case of Zeiss confocals. They have 32 detectors each, and each is looking at 9 nanometers spectral range. Now we're coming to the most important part of a microscope, and that's the objective lens. So that's really something which is very critical for the quality of the images, and it's also correspondingly expensive. So there are a lot of things which are written on an objective lens. So some of which are important are, for example, aberration corrections. So this refers to the correction for chromatic aberration. So that means how well different colors will overlap in the image. And there are different levels of the correction. Achromat, this is the lowest end. Fluor, fluotar, semi-apochromat, that's the middle range, and apochromat, that's the best correction for chromatic aberration. A very important number is working distance, which refers to the distance between cover slip, if the objective is meant to be used with a cover slip, or whatever glass it's meant to be used with, and the focal plane. So that tells you how deep into the sample you can reach. So you can see that some objectives, like those high magnification objectives, typically have very short working distances. So you can't use them, for example, with a plastic dish, because typically the plastic bottom of the dish is thicker than the working distance of such an objective. So you won't be able to reach the sample. Numerical aperture, that's a very important parameter. We'll talk about it now quite a lot. You will see that this is probably the most important, one of the most important numbers on the objective. And this refers to the basically to the width of the cone of light which can be collected by the objective. Why is that important? Well, okay, here I wanted to show you the examples of few objectives here on one of the microscopes. So here you see the working distances. So there are some objectives, those lower magnification typically have quite long working distances, while the some of the higher magnification, they have here the letters LD, which means long distance. So they have a relatively long working distance, while the oil objective has a very short working distance. And this is the CG is the thickness of glass which is meant to separate the sample between the sample and the objective. So some of the objective have correction ring, which allows to, so this is something like this. So you have a ring which you can turn, and there's a scale. So that allows you to correct for different thickness of glass. So that's why there's not one thickness, but there's a range. It's quite good to set the color according to the thickness of the glass you are using, because it improves quite a lot the quality of the image and also the light collection efficiency of the objective. So back to numerical aperture. So definitely the larger the better. That's the simple rule about numerical aperture. Why is it so? So first of all, the larger means that you can the cone of light is broader, so you can collect more light. And since the light represents your signal in optical microscopy, so it's always better to collect more light. And it also improves the resolution. So the higher the numerical aperture, the better the resolution of the objective. That comes from that there are several resolution criteria. This is which are approximately come to that the resolution. That means the smallest distance between two lines which you can resolve is approximately half of the wavelengths divided by numerical aperture, which typically translates to something for most objectives, something 200, 300 nanometer. And another way of looking at it, the resolution is determined by related to a point spread function, which is basically what would be the image of a single point. So if I have a single, let's say, single point source of light, and I image it by my objective, so then the image will be broadened. So the point spread function is basically how the image of a point source would look like. And of course, because it's broadened, so each point is slightly smeared by the point spread function, so that limits the resolution. The numerical aperture also depends on the immersion, on the refractive index of the immersion medium. That is simply because if I have different refractive indices here, so the rays of light will bend. So that's why the refractive index will influence the effective cone of the effective angle of the cone of the light. Now that we mentioned the immersion medium, so that's also written here on the objective. And one thing which is good if the refractive index of the medium matches the sample. Why is it good? It's good especially if I want to do anything like 3D imaging, because the mismatch in refractive index between the immersion medium and the sample will result in distortion in 3D. I won't go into any extensive explanation. Just I show this picture which illustrates it quite well. If I have two media of different refractive index, you can see that obviously there is a distortion in 3D. The distances here are affected by the mismatch of the refractive indices, and the chopstick looks bent, which of course it's not in reality. And another very serious thing is the degradation of the point spread function. So that means that basically the focusing of light also will be affected, and this will result in also in the loss of signal. So I will illustrate this by a little bit more microscopy related example. And here I compare two lenses at the same microscope. One is a 63 times water immersion lens, and one is a 63 times oil immersion lens. And the sample is an aqueous solution of a fluorescent dye, a very simple sample. Well, of course, the refractive index of the sample in this case is about the same as water. So it's quite different from the oil. So you see that very close to, so here, of course, this sharp rising edge, that means I come from the cover slip into the sample. And then so at very close to the cover slip, the oil objective has an advantage because it has higher numerical aperture. But when I'm going deeper, the water immersion objective performs much better. So you can see that if you are, let's say, you are looking, for example, at cell monolayers, adherent cells, so you are within 10 micrometers from the sample, oil objective might be a better choice. But if you want to look at something larger, where you want to go deeper, like 50 to 100 micrometers into the sample, then, and your sample is something which is in a water-based medium, then a water immersion objective would be a much better choice. Back to numerical aperture and to the question of resolution in optical microscopy. So as we said, it's limited to something like 200, 300 nanometers. There are so this works for, let's say, the standard microscopy, the situation which was studied by Abe when he derived the resolution criterion. But there are, of course, the situations where this doesn't hold and where a better resolution can be achieved. These are often called super resolution microscopy. I don't like this name too much because it sounds a bit like supernatural things. And one class of these techniques are called single molecule localization microscopy, which is works on a very simple principle. So I will show you, of course, this is not a real, these are simulated images, but I have here, I simulated, I have such a two concentric rings and I place fluorophores, I label them by fluorophores. So if I take an image with a 200 nanometer point spread function, so normal optical microscope, I can't see that there are two concentric rings. I can see just one big blob of light. If I could have 20 nanometer point spread function, which I can't, I would see something like this. But I can do the following trick, that if instead of all these fluorophores shining at once, most of them are dark, only with for a small for a short time, some of the fluorophores will switch on and emit light, and then they go back to dark state. So I will see effectively maybe just one fluorophore or just two fluorophores at a time from this whole structure. So I can capture such an image. And because I assume that I see just one fluorophore here, so I can pinpoint quite accurately where's the center of the point spread function. Then I take another image, another fluorophore is on in this case, and I can locate where this fluorophore, where is the center of this blob of light, and that's where I assume the fluorophore would be. And I can do this with quite high precision depending on the signal to noise ratio. I might have something like better than 20 nanometer localization precision. So then I take many such images, and I, from each of the images, I take the position of the fluorophore that was on at that time, and then I can reconstruct an image which looks like this. So I can get about 20 nanometer resolution, but it is quite lengthy, the experiments, and usually to make the fluorophores blink in such a way that they are mostly dark and on only for a short time. So only few fluorophores would be on at a time. I need special buffers, or I need special fluorophores. Another approach is structured illumination microscopy. So these two approaches can be done on one of the microscopes in Celsi, the microscope called ELIRA from Zeiss. Structured illumination microscopy is easier to do, doesn't really require a special sample, only you need to have quite bright fluorescence, but it only gives you about 2 times resolution improvement. So instead of 250 resolution, you might get something like 120. And then there's a variety of other approaches which allows you to improve the resolution. There are techniques like SOFI, SURF, that's something which Yongli has successfully been doing in some projects. Or there are techniques using artificial intelligence to process images and derive images with better resolution. Deconvolution, image deconvolution also could be part of this. They typically don't require any special sample. They can be acquired images on most microscopes, and depending on the type of the technique, you might get even better improvement than SIM, possibly, or similar. It's just an explanation how SIM works, not an explanation, just to give you an idea. So you see here a fine meshwork. If I reduce the resolution to simulate the point spread function, I don't see anymore the mesh. But if I put two meshes over each other, then because of the aliasing effect, I can see something called moire fringes, and they have larger spacing than the original mesh. So if I know the mesh which I overlaid over the original one, I am in principle able to reconstruct the original mesh from the moire fringes. And now another very important thing. So there are how the image is formed. So there are two basic strategies. One simpler is called wide field microscopy, where I have my sample, I illuminate the whole sample, and I collect the light by the objective, and that creates an image, a magnified image of the sample, which is then captured by a camera. One of the problems here, you can see that, of course, the light which comes from different points in the focal plane will focus to different points in the image plane. But the problem is that if I have light which comes from out of focus, it will reach my camera and will create a defocused blur in the image. So then I might have images which have quite low contrast of the in focus features because of the background from the out of focus features. So I would like to get something like this, where I can see with high contrast the features which are in focus. And one way how this can be achieved is by using a confocal microscope, which differs from the wide field microscope in one important thing, that it's using a small aperture called a confocal pinhole. And you see what happens. So if I have the light which comes from the focus, that will be focused in the plane of the pinhole. So it can all go through the pinhole and reach the detector. The light which is coming from out of focus will be defocused in the plane of the pinhole. So only a small portion of that light can go through the pinhole. Most of it will be blocked here. So that's how I can block the out of focus light. Of course, there's one price which I have to pay for that. And that is that if I have light which is coming from the focal plane, but from different points in the focal plane, well, that will be focused also at different points in the plane of the pinhole and won't pass through the pinhole. So I'm effectively collected light only from, not from the whole plane, like in a wide field microscope, but only from one small spot. So I don't need a camera. I can use a point detector in this case. And I don't need to illuminate the whole plane. I can just use a focused beam to illuminate the one spot from where I can collect the light. So that means that if I want to construct an image, I need to scan the focal plane with the beam. In a laser scanning confocal microscope, this is done by scanning with one point at a time. There are other versions, a spinning disc confocal microscope, where I have multiple pinholes, and that means I am collecting from multiple points in the focal plane at a time. So instead of a point detector, I'm using a camera, because then I'm capturing simultaneously the light from multiple points. So one of the the reason why confocal microscopy is done is the optical sectioning, which means I collect light only from a section which is 500, 800 nanometers thick in the sample. And another thing interesting about confocal microscopy, that it is convenient for all sorts of microspectroscopies, because I'm collecting basically the light from a single point, means single pixel at a time. So if I do any sort of spectroscopy on the light which I have collected, so I can assign I can assign get basically the spectrum for each pixel. And the question how big this pinhole should be, and the usual selection is what is called 1 area unit, which is the width of this central peak of the point spread function. So if I make the pinhole bigger than this, I don't gain much signal from focus, because you see that most of the light from the focus is concentrated in the central peak, but I will let in more of the out of focus light. On the other hand, if I make the pinhole smaller, I will improve resolution, but I will lose a lot of signal. So it's usually that is also not used frequently, because typically there's not enough signal to waste. There are alternatives to confocal microscopy, how I can do optical sectioning. So one is structured illumination, also has this sectioning ability. There are things like deconvolution or artificial intelligence processing. So again, these computational approaches. And another thing which is very similar to confocal microscopy, in the sense that is also a point scanning approach, is multiphoton microscopy. So that is something we don't have at cell C at the moment, but it's something that is in plan to install. So maybe in the future we can talk more about this. Another approach is total internal reflection fluorescence, which is a form of sectioning, but not, I cannot do arbitrary sectioning. I'm only looking at a section which is close to an optical interface. That means close to cover slip surface. Now I'm using phenomena of total internal reflection, which means that light which hits the optical interface at a large angle is reflected completely, but part of the energy still can penetrate over the interface and can excite fluorescence in a very thin layer of the sample, which is usually about 100 nanometers. So this is often used to image cell membranes or things like that. Just for fun illustration that of total internal reflection. So I have here, there's a sticker, and I put a cup over it, and I can see the sticker, of course, because the cup is transparent. Then I pour water into the cup. And now, because there's an optical interface, because there's air, the sticker is in air, but there's water inside of the cup. So I have total internal reflection on this surface, on the bottom of the cup. So when I'm looking from here, I cannot see the sticker. But of course, if I look from the top, not under the angle, I can still see the sticker, because the total internal reflection happens when for rays which hit the interface on the larger than critical angle. You can see that actually when I'm looking from top, I can see the sticker better with the water inside the cup than when there's no water. So this is, again, an example how immersion liquid helps in microscopy. And the last important topic is image sampling. Let's start to explain what we mean under image sampling. So I have my object, and I create a magnified image. So let's say in this case I have done 10 times magnification. Again, in case it's not clear, this is not a real microscopic image. I haven't taken a small cat, which I would image under a microscope. I don't have such a cat, because they are not allowed at HDBs. Let's say my resolution is, so this would be the ground truth, but let's say I have some limited resolution. So I have 300 nanometer resolution. So that means I cannot resolve all details. And now, of course, this would be the image created by the objective. But then I capture the image by a camera, and the camera has a certain grid of pixels. It's a camera chip. So let's say my camera has 36 micrometer pixel on the chip. So of course I cannot I cannot resolve anything which is smaller than the pixel. So then my my sampled image will look like this. So through the sampling by the camera, I have obviously lost a lot of details which were present in the image created by the objective. So I need to obviously I need to have a camera with finer pixels. So here the pixel in the sample space, in this case, because I have 36 micrometer is the size. So so that corresponds in the sample space to 3.6 micrometer divided by the magnification. So that, and you see that this is obviously much larger than the resolution. So that's why I am losing resolution by the sampling. If I take a camera which has smaller pixels, then the pixel in the sample that would correspond to 140 nanometers in sample, and that is smaller than the resolution. So I'm not losing any details through the sampling. I should have the size of the pixel in the sample space should be smaller than 1/2 of the resolution, so that I don't lose any detail. What if I take a camera with even smaller pixels? Would I gain anything? Of course not, because I've been already limited by the resolution of the objective. The only thing the only change that it can cause is that I will divide the signal between more pixels, so I will have less signal in each pixel, and typically the camera will have some readout noise, which is signal independent. So the result will be worse signal to noise ratio. So that's why it's better not to oversample the image too much. With a wide field microscope, of course, I don't. I mean, the sampling is defined by the size of the camera pixels. So there's not much I can change unless I reconfigure the microscope. But typically, this is something which is done when the microscope is set up. So the choice of the objectives and the camera should be done in such a way that there is a reasonable sampling for all objectives. Because in general, the numerical aperture and the resolution of the objective decreases with the magnification. So in that sense, the same camera gives a reasonable sampling for different magnifications. Of course, if I have two objectives with the same magnification, but different numerical aperture, as is the case here, we have 263 times objectives, then of course I can't have, then one would be either undersampled or the other is oversampled. I can't avoid this. So in this case, if I use this the resolution criterion, which means that the resolution is half of the wavelengths, can use 550 as the middle of the visible range divided by the numerical aperture. So you can, and this microscope, so for this microscope, actually, this one is about the ideal sampling. So for this one, I will have an oversampled image. Now the situation is very different for a point scanning confocal microscope, because here I don't have a camera, but the scanning scanning is defined by the sampling is defined by scanning format. So so obviously it's it's a different situation if I do scanning like this or like this. So how many lines I scan the area and with into how many pixels I divide each line. That determines the sampling. So I'm not limited by any camera. I have the absolute freedom to choose my sampling. So the question is how to choose it. Of course, I can look at the numerical aperture of my objective and calculate what is the resolution, and according to that determine the pixel size. But the manufacturers of the microscope make our life easier. So typically there's always some button which says something like optimize format. So if you press this button, it will set automatically how many pixels and you need to have properly sampled your field of view. Now the magnification in a point scanning microscope is defined by the sweep of the scanner, which is usually what is called the zoom in the software. So it's obviously a different situation when I scan like this, or if I scan like this. Because scanning like this, it means effectively I'm zooming into a smaller area in the sample, so I'm getting a more magnified image. Obviously, the important thing that if I change the zoom factor, that means I change the scanned area, I need again to optimize the format. So when I change the zoom, then I again should press the optimize XY format button so that I keep the same pixel size. Otherwise, the images will not be consistently sampled, and it will not be possible, for example, quantitatively compare the intensities in the images. Now, what is the role of objective magnification in confocal scanning? So the objective magnification defines the maximum area which I can scan. So the question is, so since the resolution is determined by objective numerical aperture, so the question is, if I have a choice of a 63 times 1.4 numerical aperture or 100 times 1.4 numerical aperture, which one is better? Definitely the 63 times. Why? Well, because they both give me exactly the same resolution, but the 63 times allows me to scan a larger area. So the 100 times doesn't give me any advantage at all. The other thing, so you may say, okay, but also if I'm not interested in a large area, then it doesn't matter really. I can take the 100x. Well, actually, even then, if you don't utilize the full range of the scanner, the 63 times has an advantage because the objectives work the best for arrays which propagate along the optical axis. If I increase the angle of the ray, the more larger the angle, the worse will any residual optical aberration become. So since the 63 times, if I want to scan the same area, can do it with the ray going under a smaller angle. So that again makes the 63 times a better choice. So if I have a choice of such two objectives, definitely always choose this one. You can see it even has a longer working distance. So there's really so if you ask if there's any advantage of the 100 times, there's no advantage at all. And then you may ask why such an objective is there. The answer is it's there only for historical reasons, because I'm afraid to take it out, because there might be some user, unfortunate user, who started using the 100 times objective, and for the sake of consistency of their data, they should continue with it, even though it's a bad choice. If I was at the moment here, at the moment when the microscope was commissioned, I would never allow the 100 times objective to be placed there, because the only thing it does, it misguides users to using a suboptimal objective. So if you are starting, if you don't need to use it for the sake of consistency, don't use it. That's the important take-home message. So now, some bits and ends at the end. Of course, we also have equipment for environmental control. So there are some microscopes which are in heated enclosures. So you can, if your sample needs 37 degrees physiological temperature, you can use such an enclosure. There are also stage top incubators. So you can put that on the stage of any other microscope which doesn't have the enclosure and maintain the temperature. And also you can connect CO2 cylinder to this unit if your sample needs 5% CO2, for example, for long-term time-lapse acquisitions. Also, there are some microscopes which have hardware autofocus, which is again very helpful for long-term observations, because without that the focus might drift over time, so you will lose your focus. But with a hardware autofocus, you will remain focused no matter how long your experiment lasts. And there are, of course, besides the actual imaging, there are all sorts of other things that can be done with the microscopes, for all sorts of microspectroscopies. For example, we can measure how fast do molecules move by techniques such as FCS, FRAP, fluorescence recovery after photo-bleaching, single particle tracking, identify molecules or certain chemical bonds in molecules using Raman microspectroscopy or Brillouin microspectroscopy to measure stiffness of materials. So these things can be done. Of course, these are more difficult things to do. So if you think that this is relevant for your project, then approach Yongli or me, and we can discuss how this can be done and help you with these experiments. And the last, very simple summary. So there are different aspects of an imaging experiment which I want to say maximize. I want it to be fast. I don't want to waste time on the experiment. I want to get the best resolution. I want to get the best sensitivity. That means to have to capture even weak signals, but I can't maximize all of them at once. So that's why they are drawn at the points of a triangle, because if I maximize sensitivity and resolution, I have to sacrifice the speed. So I have to, there's not one setting which will work for all. Different experiments might have higher requirements either on sensitivity or on speed or on resolution. And that's why I need to search for such position in this triangle which will work the best for my experiment. For live cell experiments, I even might add another dimension to that, to be live cell compatibility. So then it's no more just a triangle, it's a tetrahedron. And there's a summary which maybe Young we can comment on this, because this is a chart which he created, which is a sort of microscope selection guide.
Yeah, so basically, this is a brief guide for users who wants to choose the microscope. Obviously, you start from the top. You look at whether you need an upright or an inverted. That's basically based on your project. Whether you're growing cells on a dish, you're looking at a tissue on the slide, right? And then you have to decide whether you are using just the transmitted light, which gives you a certain contrast for your project. But of course, if you need even higher contrast from the fluorescent, you will most likely be using the fluorescent techniques. The basic one, which is the wide field and the confocal. As Vedek has already pointed out, there are other methods. For example, if you want to increase the resolution, you can use the super resolution microscopes, the same palm and the stone. You also need to think about whether are you just taking the static image? Are you tracking proteins or looking at fast dynamics? Are you looking at sections? If you're looking at sections, maybe confocal. Are you going to do Z-stack like doing a 3D reconstruction, or looking at certain parts of the cell like on the membrane? All these questions that you want to ask leads to the proper fluorescent techniques that or the microscope that you probably will need to use. And of course, you need the temperature, long time lapse, for example, or CO2. And after choosing or deciding which microscope you probably will be using, then of course you have to be at least a bit clear about whether which objectives that you might be using. So you have to consider about the mainly, of course, the magnification will give you either a big picture or are you looking at small details. If you are going to zoom it all the way in, and of course you need to consider about the resolutions that you need. Are you going to look at single proteins in a cell, or basically are you just looking at a single cell, right? Then after that, of course, we covered the pixel size, the proper sampling. You have to take note of your pixel size, because I encountered a lot of users who sometimes after a few experiments, they tweak some stuff and it messes up their pixel size, and they don't really know until months later or when they are about to publish. So please take note of the users, the pixel size every time when you go onto the microscope. Of course, at the end of it, which we did not cover in this orientation, is you have to consider where you store your data, as well as the post-processing, the image processing of your data. Right, so basically that's it. So I'll leave it to Vedek to do the final conclusion.
Okay, thank you, Gui. I would add here to the last point, the image processing. So of course the experiment doesn't end with acquiring the image, and there's a lot more work that can be done with the images. I already mentioned that there are things to let's say improve the quality of the images, so things like deconvolution or any of these new AI image processing methods. But there are of course a lot of other things, how to analyze the images and derive more information from them. And we can, of course, also, we are helping some users with image processing, so you can approach us if you need any help on that side. And connected with that is that we are planning to make an introductory course on imaging Fiji. And I have done in my previous job a similar course, so it was quite popular there, and it lasts it was roughly 4 hours. So there I did it in one shot. So it was like a whole afternoon from 1 p.m. until evening. But I don't know if we are now thinking what will be better for preferred for you, if to do it in one shot or to do it in, let's say, 3 sessions. So for those who are here, you can maybe type to the comments if you have any preference on one or the other format, so that we can decide how to run it. I think that's all from me. And if you have any questions now, we can. Okay, people are preferring several sessions, so I think this is good that there is one of the options which seems to be preferred by multiple people. Thanks for the feedback on that. And now, if you have any, definitely it contains practical examples. That's clear. And when we did that, I always asked people to bring their own. At that time, there was no COVID, so we of course did it in person. Now it probably will be better to do it via Zoom, which means that people will be sitting at a computer anyway. So when we did it in person, I asked people to bring their laptops so they can try directly the things in MHA as we were showing the examples. Okay, if you have now any questions to what we were talking about now, you can also ask. I don't see any questions coming. So I would say that since there are no questions coming, I think we have exhausted what we wanted to say, and we have exhausted the time we have allocated for this session. Uh, so thank you all for joining us, and I think can gradually close this.
Thank you all for coming.